Hello everyone, my name is Henry, and today our group will present to you the fat-soluble vitamins A, D, E, and K. You probably have the general idea of what fat-soluble vitamin group is, so instead, we will tell you the important and interesting things from each specific vitamin that we have learned through our research. Without further ado, let's begin with vitamin A. On the topic of vitamin A, Alexandra and I will walk you through the properties, functional roles, natural sources, the synthesis process, and why the bottle of vitamins you got from Costco is so expensive. So let's dive right in.
So first off, I'm going to talk a bit about the main structure of vitamin A. So as we all know, vitamin A is a group of fat-soluble vitamins, which can be available in the human diet in two main forms. Retinol, which is found in animal-based foods, and provitamin A, which is found in plant-based foods. The most important provitamin A carotenoid is beta-carotene, which can be processed by the body in several ways. So as we see right here, we have the molecule of beta-carotene, and that molecule contains two beta-ionic rings on each end of the molecule. Those rings are linked by a polypenoid chain over here, and that chain can be cleaved enzymatically using oxygenase, which will give rise to retinol, which is an essential vitamin A. And from one molecule of beta-carotene, you can produce two molecules of retinol. So next we're going to look at the different forms of vitamin A. First off, we have retinol right here, which contains the alcohol group. Then we have retinol, which contains the aldehyde group. And finally, retinoic acid, which contains a carboxylic acid group. They can each be converted in our body in different ways. Retinol has the ability to convert itself to retinal, and retinol has also the capability of converting itself to retinol. And therefore they're both interchangeable. Retinol has also the ability to convert itself to retinoic acid. However, it's important to note that it is only a one-way reaction, and retinoic acid is unable to convert itself to retinol. Next off, we're going to look at the role as well as the importance of vitamin A consumption. So retinoids are very important for growth, bone maintenance, reproduction, have very important antioxidant properties, as well as immunity functions. Specifically, retinol is important for vision, and retinoic acid helps in cell differentiation. Vitamin A deficiencies are very rare in developed countries, but the most important concern with that deficiency is blindness, as well as the inability to see well in the dark or in low light. However, an excess may result in headaches, nausea, dry and cracked skin, as well as liver disease. Alright, so next we're going to look at the natural sources of vitamin A. I just want to quickly mention first that for individuals that consume animal-based food products, deficiency concerns are actually relatively low. Concentrations of preformed vitamin A are highest in liver and fish oils, but you can also find that in meat and dairy products, yellow or orange fruits, as well as in green leafy vegetables. Research suggests that vitamin A found in fruits and vegetables are mainly in the carotene form and may not be as well absorbed as the vitamin A found in animal-based food products, known as retinols. This is simply due to the fact that they are both metabolized quite differently, and their absorption is also different because of that. So just to put this into perspective, you would have to eat 4 pounds of carrots in order to get the same amount of vitamin A as in 3 ounces of beef liver.
Like Alexandra mentioned, deficiency of vitamin A is pretty uncommon in developed countries like Canada and the United States. And this is possible due to the fortification of vitamin A in our diet, usually in common food like milk, cereals, grains, and oils. This ensures that the majority of population, even though they don't eat a lot of vitamin A rich food on a daily basis, we still get the necessary amount to prevent deficiency from happening. And so I'm going to walk you through one of the synthesis process of vitamin A that's been used commercially. We begin with the basic compound that you see right here, that is beta-ionol, the backbone structure of vitamin A. Then we have the first reaction, which is a chain addition of 3 carbons, make it a total of 7 carbons. See 1, 2, 3, 4, 5, 6, and 7. The next reaction is also another chain addition. This time it's a 4 carbon, making it a total of 11 carbon chain. Then we have the hydrogenation of the triple bond into a double bond using palladium as a catalyst. Otherwise, the reaction is going to continue forward to make it a single bond, which is not what we want. The next step is to remove the OH group by dehydration. And because of the removal of a single bond, we have a double bond being formed on the chain. You can see right here. And due to the potential isomerization, we have a lower percentage yield compared to other steps in the process. Then we have the conversion of the alkoxy group, you see right here, into an aldehyde. And because of the change in structure, we also have the rearrangement of all the double bonds that you see right here, so that they can achieve a stable conformation. The next step is to isomerize the location of the double bond in the carbon ring. You see right here. So this makes the compound into retinol, which is the aldehyde form of vitamin A. The final step is to convert the aldehyde into alcohol, which is the retinol that we want. This is possible by a reducing agent that you see right here. And if you pay attention to all the percentage number that we've seen so far, the end product usually is about 37 to 38% of what we begin with. So this explains why when you go to Costco and you get a bottle of vitamins or supplements, usually they are more expensive compared to when you go and get some carrots, for example. Because there's so many potential isomers of vitamin A and other byproducts that need purification, you want to make sure that the one that the customer is paying for is actually what they're getting and not its brothers and sisters of isomers. Because they will have a much lower efficiency compared to the pure form of vitamin A. Sometimes they may have side effects as well. And so this also wraps up my part on vitamin A. And so I'll pass it along to Wenya and Nancy so that they can continue with vitamin D. So take over, guys.
Vitamin D is a fat-soluble vitamin which has a special cyclosterol structure. Cycle means cut in Latin, thus cyclosterol means a sterol that has been cut open, which we can see a clear difference in structure between provitamin D3 and vitamin D3. Recommended dietary intake for vitamin D is 600 IU for most people, while infants need less and seniors need more. UV biosynthesis in our skin is the major vitamin D source for humans. Therefore, due to lack of UV exposure in Canada during winter, vitamin D deficient is a common problem for Canadians. Thankfully, a few dietary sources also contain significant amount of vitamin D that can help Canadians against vitamin D deficient. There are a total of 5 types of vitamin D molecules. Among them, D2 and D3 are most important ones because D3 is the type that produced by our skin, and D2 is the one that commonly produced in commercial means as either fortification additive or dietary supplements. The key function of vitamin D is helping human body to absorb minerals like calcium, magnesium, and phosphorus, which its mechanism will be introduced in the next slide. In our body, vitamin D is first being metabolized into 25-hydroxy vitamin D in the liver, then into 125-dihydroxy vitamin D in kidney. This metabolized form acts as hormone to tell organs like kidney, intestine, and bones to either absorb more minerals or to excrete the extra minerals. Therefore, maintains the mineral homeostasis of our body. Since minerals like calcium, magnesium, and phosphorus is the building are the building blocks of our bone tissue. Therefore, vitamin D deficiency usually leads to bone stress and the development problems like osteoporosis and rickets. In terms of vitamin D application, the most common application of vitamin D in the food industry are nutrient fortification and supplements, since it plays such important role in human body's mineral homeostasis and bone health. Recent research also revealed other potential applications of vitamin D, like reducing risk of colon process and breast cancer, fight against inflammation, as well as preventing and treating insulin resistance for diabetes patients. Besides UV exposure, some food can also be a good vitamin D sources when UV exposure is not readily available. There are mainly fatty fish like cod and salmon, especially cod liver oil, which we can see its content in the table shown. While vitamin D fortified foods are also good choices, like other fat-soluble vitamins. Vitamin D dosage also leads to toxicity. Chronic symptoms including kidney stones and irreversible calcification damage to soft tissues like blood vessel, and the acute intoxication usually results in hypercalcemia, which is high blood calcium level.
Since vitamin D isn't found in many food products, some countries will benefit from direct supplementation or fortification in specific foods. This can be especially important in places where it doesn't shine all year round, such as Canada, where winters are particularly long and about 40% of Canadians are actually deficient in vitamin D. Deficiency can also be more prevalent in people with darker skin because they have higher levels of melanin, which is a natural protectant from the sun, and which means the UV rays are not as readily absorbed by their skin, which can hinder vitamin D synthesis and cause several health issues. As mentioned previously, there are two main forms of vitamin D which are most important for humans, vitamin D2 and vitamin D3. These are the main forms that we will find in supplements and fortified foods. Vitamin D3 is usually made from an animal source called lanolin, which is a waxy substance that is secreted by the skin glands of sheep to moisturize their wool, but it actually also provides the chemical precursors for vitamin D3. So the extraction process is somewhat simple. First, the wool is going to be washed to remove any undesirable products such as dirt, feces, and parasites. Then the wool is washed once again, but with hot water and detergent to remove more impurities, but mostly to extract the crude lanolin. The crude lanolin will undergo centrifugal separation or solvent extraction so that we can obtain the wax, which will be used in many different ways. So for example, in the food industry, it undergoes UV irradiation to make vitamin D3, but it could also be used in the cosmetics industry since it has many waterproof and barrier qualities. Now just to quickly touch on vitamin D2, it's usually made from UV irradiation of ergosterols, which are naturally found in yeasts and are more suitable for people who wish to avoid animal-based products. Now, since vitamin D is subject to so many manufacturing processes, it's also going to be subject to loss in potency. One of the ways it can lose some of its activity is through iodine, which is commonly added as a chemical in foods or pharmaceutical products. It can also be found in cleaning and sanitizing products, which are used in the food industry. Even in very small concentrations, iodine will cause the cis form of vitamin D to be isomerized to its trans isomer, which leads to its degradation and the loss of its health benefits. Vitamin D is also susceptible to a low pH. So in acidic conditions, vitamin D is going to be isomerized to isotachysterol, which can be useful in identifying vitamin D compounds in different substances. However, it is very unstable and rapidly oxidized in the air at ambient temperature compared to vitamin D3, which is relatively stable in the same conditions. Finally, vitamin D activity can also be reduced or lost through cooking processes such as boiling, frying, and baking.
So now we're going to be talking about the fat-soluble vitamin, vitamin E. I will be presenting on its structures, functional roles, and natural sources, and my colleague Mandeep will talk about its uses in the food industry and the effects of processing. Vitamin E was first discovered in 1922 as a nutritional factor that led to infertility in lab rats. Today, vitamin E is a collective term given to a group of 8 different isomers of fat-soluble compounds. Naturally occurring vitamin E includes alpha, beta, gamma, and delta tocopherols and tocotrienols. All forms, both tocopherols and tocotrienols, share the same basic chemical structure, which is characterized by a long isoprenoid side chain attached at the second position of a 6-chromanol ring. Where the tocopherols and tocotrienol groups differ is in their level of saturation in the side chain. So on the left, you can see that tocopherols contain a saturated phytyl tail in their side chain, whereas on the right, you can see that tocotrienols contain 3 double bonds in their side chain, so it is unsaturated. Among the alpha, beta, gamma, and delta molecules, the number and position of methyl groups on the chromanol ring differ. As you can see in the figures, the chromanol ring is trimethylated or fully methylated in both alpha molecules. In the beta and gamma molecules, the chromanol ring contains 2 methyl groups, so it is dimethylated, and in the delta molecules, the chromanol ring is monomethylated. Among all of these, alpha tocopherol is the most active, and of the tocotrienols, only alpha tocotrienol has significant biological activity. So vitamin E isolated from natural sources has the R configuration at all 3 asymmetric centers. So it consists of just one isomer. RRR alpha tocopherol is the most biologically active molecule, and natural vitamin E is labeled as D alpha tocopherol in supplements. So while natural tocopherols have just one isomer, the synthetic production of vitamin E results in a racemic mixture that contains 8 different isomers as listed on the bottom here. Of which only 12.5% of the racemic mixture is represented by RRR alpha tocopherol. Synthetic forms of vitamin E are labeled with a DL prefix on supplements. So in the body, vitamin E acts as an antioxidant, helping to protect cells from the damage caused by free radicals. The body also needs vitamin E to boost its immune system to fight off invading bacteria and viruses. It helps to widen blood vessels and keeps blood from clotting within them. Vitamin E deficiency is rare and occurs almost exclusively in those with an inherited condition that impairs their ability to absorb the vitamin, such as cystic fibrosis. Deficiency also occurs in those who cannot absorb dietary fat or have rare disorders of fat metabolism. Deficiency causes nerve and muscle damage that results in loss of feeling in the limbs and body control movement, muscle weaknesses, and vision problems. In terms of excess, eating vitamin E from foods is not risky. In supplement form, however, high doses of vitamin E might increase the risk of bleeding by reducing the blood's ability to form clots after a cut or injury, and it might also increase the risk of serious bleeding in the brain. Lastly, vitamin E is obtained exclusively from the diet from fat-containing foods. Nuts, seeds, and vegetable oils are among some of the best sources of alpha tocopherol, and 1 tablespoon of wheat germ oil provides 100% of your daily value.
So next is the uses of vitamin E in the food industry. Vitamin E acts as antioxidant. So due to this property, it is used in the vegetable oil because oil are very prone to oxidation. So to prevent the oxidation of the oil, vitamin E is intentionally used in the oil. So vitamin E is also used in the fortification of the beverages, but vitamin E is not water soluble. Hence, it is used in the form of nanoparticle emulsions, which stabilize the 2 immiscible liquids. Vitamin E also prevent or stabilize the red color of the meat. The loss of red color is due to the oxidation of the red oxymyoglobin into the brown metmyoglobin. So when the vitamin E added as a supplement in the diet of animal, it prevents the red color of the meat. Um, Due to the degradation of the polyunsaturated fatty acid, there is a formation of the short chain compounds like ester aldehyde ketones, which gives off flavor in the meat and meat food products. So vitamin E prevent this degradation of the polyunsaturated fatty acid and also prevent the off flavor of the meat products. Vitamin E is also prevent the drip loss in meat, which is a liquid comes from the membrane of the meat because of the effect of the oxidation on the semi-permeable membrane of the meat. So so there is a leakage of liquid from the membrane of the meat, which is not desirable, and vitamin E content prevent this drip loss in the meat. Vitamin E also prevent the cholesterol oxidations. And next is the processing effect on vitamin E content. So when refining of oil occur, there is a loss of vitamin E content. So here are some oil in which refining affects the vitamin E content. So refining of oil include different steps like degumming, bleaching. So all step has some effect on the vitamin E content. So so during refining of the olive oil, the 15% of the vitamin E content lost. In the refining of soybean oil, 25% of vitamin E content lost. And in same, the rapeseed oil, 32% of vitamin E content is lost. In corn oil, 35 to 40% of the vitamin E content is lost. So the refining affects the vitamin E content. Then what are the effects of the cooking on vegetables? So in some cases or in some vegetables, the cooking increase the vitamin E content, or basically the tocopherol content of the vegetables. Like in broccoli, spinach, perilla leaf, cooking increase the tocopherol content of these vegetables. Or on the other hand, in potatoes, sweet potato, carrots, cooking decrease the tocopherol content. Then here are some different different food process which ultimately reduce the vitamin E content or tocopherol content, like milling and bread making reduce the vitamin E content. Bleaching of oil is also reduce the vitamin E content. Then the microwave heating of seed oil decrease the tocopherol content. Irradiation of rice bran reduce the vitamin E content, and when sterilization of the diet also affect the tocopherol content and reduce the vitamin E content. Before ripening, the bagging of the fruits also decrease the vitamin E contents. Thank you.
So now we are going to be talking about the fat-soluble vitamin, vitamin K. I will be talking about the structure, functional roles, and natural sources, and Emily will be talking about the synthesis and processing effects on the vitamin. So a bit of a background on vitamin K. It is involved in coagulation, bone development, and cardiovascular health. There are 3 known forms of vitamin K. The first 2 forms are the primary forms, and they're known as vitamin K1 or phylloquinone, and vitamin K2 or menaquinone. The third form is a synthetic form known as vitamin K3 or menadione. If you take a look at the structures of the different forms of vitamin K, you'll see that they all have the same backbone, which is a 2-methyl-1,4-naphthoquinone. But they have different substituents on the third carbon of the naphthoquinone ring. So K1 substituent is a phytyl chain on the 3' position. K2 has an isoprenoid side chain, a varying number of isoprene units, and K3 has no organic moiety on the 3' position. Now let's take a look at the different roles, deficiency, and excess. Vitamin K1 aids in blood clotting. Vitamin K2 is involved in calcium transport, and it prevents calcium deposition in the lining of blood vessels. It also helps improve bone density. K3 is currently banned by the FDA in the US because of potential toxicity, but it is being studied as a potential hepatocellular prostate cancer therapy or as a potential treatment for skin toxicities. Observed signs of vitamin K deficiency are significant bleeding, poor bone development, osteoporosis, and increased cardiovascular disease. When vitamin K is taken in excess, toxicity can occur and it manifests with signs of jaundice, hyperbilirubinemia, and hemolytic anemia in infants. For the sources of vitamin K, K1 is found in green leafy vegetables and plant oils. K2 is found in egg yolk, butter, cheese, and fermented foods. And K3 is a synthetic analog. Looking at the different processing effects on the vitamin K content, you'll see that it is relatively stable when it is heated or frozen. During cooking, there is an observed increase in the vitamin K content. The increase may be because heating causes the vitamin to be released due to the breakdown of the plant cell wall. This makes it available for detection by HPLC. At low pH, the vitamin K content decreases. When there is light, vitamin K content in oil tends to decrease. On the other hand, it increases in produce. When storing produce, the phylloquinone content increases days within storage in both dark and light stored produce. However, it increases more in light stored produce. Lastly, vitamin K content is stable with irradiation.
So a little bit about hydrogenation. It doesn't only impact the fatty acids, it also impacts vitamin K1. By saturating its double bond on its tail, it turns vitamin K1 phylloquinone into 2,3-dihydrovitamin K1, or DK. And this level of transformation is commensurate with the level of hydrogenation. The more hydrogenated a product is, the more DK there is, and the less K1 there is. In this day and age where trans fats and hydrogenation are being banned, this means that K1, or more specifically DK, can serve as a very useful indicator of hydrogenation, because as we know, not because something is banned that people will necessarily listen and stop doing it. So it's very helpful to have indicators like this that can help identify hydrogenated products. Now, food science doesn't start at the processing plant. It can start on the farm. And so I wanted to mention a couple of points about agriculture of crops in which we want to maximize vitamin K content. So for those of you who thought the great Montreal-Boston rivalry was hockey, it's actually kale. Apparently, a study was done, and kale and spinach were grown in Montreal and in Boston. And from that type of study, it was found out that vitamin K actually grows more, or there's a higher vitamin K content in cultivars grown in colder climates. Just like the richer the soil, the more vitamin K. The younger the leaves, usually the more vitamin K as well. And the choice of cultivar or of species is important. Some have more, some have less. And of course, there is always the possibility of specifically developing higher vitamin K yielding cultivars by genetic manipulation or other techniques. A quick word about warfarin, which is a pesticide, a rodenticide often used in agriculture. It is an anticoagulant and it is an antagonist to vitamin K. They counteract each other. So obviously, if you're having a kale farm, if you're, wanting to maximize your vitamin K content in your kale, you're going to want to look for a different rodenticide. And so now a mention of fermented foods as a source of K2 or menaquinone. So our gut bacteria make K2 for us in our colon, but all bacteria or some bacteria do it in external sources, which is fermented foods like yogurt, natto, and kefir. Why do we care about K2? The basic health roles that it has, but also there are lots of new scientific studies that show that it plays an important role in lots of prevalent diseases like obesity and neurological diseases. And a very recent study shows that it actually improves cognitive function significantly in the elderly. And with the aging population, that type of claim means functional superfood development. And so the food industry is looking into the development of both pre- and probiotic bacterial strains, of prebiotics and bacterial strains that maximize K2 content in food. And so an example of this is B. subtilis BCRC 14715 was studied and found to maximize K2 content in natto specifically. And for the dairy side of things, studies were conducted that show that various combinations of bacterial cultures have different effects. Some are synergistic, like those mentioned on the slide here between L. casei and S. typhimurium and B. fragilis, whereas other combinations are actually detrimental and reduce the K2 content versus single strains. And so it is the development of these cultures, of these bacterial cultures, that will maximize your K2 content that is the basis for the development of these new superfoods that I am sure you heard it here first, will be populating the yogurt aisle eventually.
Okay, so that will wrap up our part on the fat-soluble vitamins. Thank you so much for listening and attending our presentation. Have a great rest of your day. Bye!